obviously causes clotting. This is just a handful of the clots I'm still examining in the lab and waiting on a couple more reagents so I can finally publish some studies. The coagulation pathways, we know that the spike can help trigger antiplatelet factor 4 antibodies, which was much of the mechanism of clotting in some of the J&J adenoviral vector shots. That spike protein induces a fibrinogen-resistant like clot. And this is very, very important in the sense that one can take platelet-poor plasma, and I'll touch on this here in this study by Dr. Pretorius out of South Africa. I think this is one of the seminal papers of all of COVID. You can take platelets out of plasma, just put that S1 subunit into the plasma, and the proteins will clump in the absence of platelets. So platelets aren't necessary in that clotting cascade. This is critically important. And this is why we're seeing these very resistant microclots. And much of the traditional medicines we use as anticoagulants don't seem to work. And I know Dr. Corey may talk about some of the treatments, but I'm happy in the Q&A to mention some of the ways we can look at breaking some of these down. These are amyloid-like. I've done Congo red stains looking for amyloid protein. I've got another round of an amyloid marker that I'm going to try to stain these with because they didn't show traditional amyloid when I looked at the 30 clots that I received from different postmortems. So I want to look at the different protein products. So really just congealed sheets of proteins. And these microclots are not traditional in what we think of as clumps of materials. Sure, they have a lot of fibrin in them, but they're resistant to fibrinolysis. And I think there's a couple interesting mechanisms I could try to explain there. And we see these amyloid fibrin-like microclots both in long COVID as well as post-shot injury. And there are a lot of therapeutic implications here. Again, just another paper by Dr. Pretorius showing more of the same. Again, here's just a for example of what they look like. These aren't traditional clots. They're long, they're stringy, they're polypoid, and very, very rubbery in texture. They're not just post-mortem. I've been receiving recently from some cardiovascular interventionists around the country, some in vivo specimens that they've been fishing out of different vessels. So here's just another example of one that I just received fragments from last week while I was traveling. So we'll be examining this as well. And this is what they look like. On the left-hand side there under the microscope, you can see trapped within the clots are certainly many white blood cells and blood products. Those seem to have an interesting upregulation in the markers I've done so far of what we call intercellular adhesion molecules. So not only are the clots forming a thick protein, they're also upregulating the protein that helps them stick to each other, which is fascinating. In the middle in that pale area, that's that amyloid-like proteinaceous material that we're seeing. Again, why would we do this to our children? Give them a shot for a virus that's extinct.